Now, today we will discuss about transcription and we understand what is transcription. Gene transcription is a process by which information of DNA strands are copied into different types of RNA, such as messenger RNA, leading to the synthesis of protein through translation, or non-coding RNA, such as microRNA, transfer RNA, and ribosomal RNA. Both the process replication and transcription are chemically and enzymatically very similar, as both of them involves polymerase enzyme, which synthesizes a new strands of nucleic acid in 5' to 3' direction, complementary to a DNA template strand 3' to 5' direction. Transcription is controlled by protein in DNA region. These proteins can either activate or deactivate transcription. Changes in chromatin like moving nucleosomes or modifying histone are affect transcription. Now we need to understand what are the key difference between replication and transcription. When making RNA during transcription, we use ribonucleotide instead of deoxyribose nucleotide like we do in DNA replication. Unlike DNA replications where a primer is needed RNA polymerase, the enzyme for transcription can start the process on its own without a primer. The RNA doesn't stay connected to the DNA like newly formed DNA does during replication. Instead, the enzyme pushes the RNA strands a short distance away as it adds new ribonucleotide. This push action is important because it allows the RNA to be used as a template for making protein. Only a small part of the RNA stays attached to the DNA within the enzyme is activated site. As the messenger RNA moves away from the enzymes, it separates from the DNA. Only about 8 to 9 RNA nucleotides remain paired with DNA. The mRNA, now single strand, is ready for translation, which happens alongside other cellular processes in prokaryotes. Transcriptions can happen multiple times, and several RNA polymerase molecules can work on the same gene simultaneously, producing many transcripts quickly. Cell can make lots of transcripts from one gene or DNA sequence in a short time. Only a specific part of DNA are copied during transcription, about 40% of the genome, unlike replication which copies the entire genome. Replication needs to copy the whole genome once during each cell division, while transcription can make multiple copies of a section. Replication happens only once per cell cycle in eukaryotes. What are the template coding and sense strand? When DNA gets transcribed, either strands can be used as a template depending on where the promoter is. The strands of DNA that gets used as a template for the transcription is called the template strands, written to form of 3' to 5' direction. The template strand, also called the antisense strand, its sequence matches up with the mRNA that's made during transcription. The template antisense strands hold the code for tRNA anticodons except use thymine in DNA instead of uracil in RNA. The strands of DNA not being used as a template is called the non-template or sense strand. It's also known as the coding strands has the same sequence as the mRNA, but with a key difference. In the RNA, all the thymine nucleotide are replaced by uracil nucleotide. The transcription machinery starts copies DNA when it finds a special DNA sequence called promoter. Promoter are like sign for RNA polymerase telling it where to begin copying. RNA polymerase copies the DNA until it gets signals to a stop. When RNA polymerase reaches a specific DNA sequence called a terminator, it stops copying. This is called termination. Now we need to understand how accurate the transcription compared to the replication. Replication is more accurate than transcription because it makes fewer mistakes. Transcriptions has highly accurate but makes more mistakes compared to replication. Replications makes about 1 mistake for every 10 nucleotide added, while transcription makes about 1 mistake for every 10,000 nucleotide added. The reason for the difference in accuracy is that transcription lacks through proofreading mechanism. Although there are two types of proofreading for RNA synthesis: pyrophosphate editing and hydrolytic editing. Why it is more important for DNA replication to be accurate compared to transcription? DNA holds our genetic information, and DNA replication is how that information is copied. Errors during DNA replication can be really bad because they become a permanent part of the individual genetic code and can be passed down to their kids. Transcription makes temporary copies of genetic information, usually making multiple copies of each part. So, if there's a mistake during transcription, it's not usually a big deal because it just affects one of the temporary copies. What are different steps in transcription cycle? Transcription of gene can be divided into a series of 3 well-defined steps such as initiation, elongation, and termination.